Okay, good evening, everyone. I'm pleased to welcome you to the Community Science and Technology Special Virtual Seminar Series, Demystifying COVID-19, presented by Pacific Northwest National Laboratory. This session is being recorded so that those who are not able to attend today can view it at a later date. My name is Jim Sanford, and I'll be your moderator today and throughout this exciting 5-part seminar series. The seminar series is designed to share what we've learned so far and answer questions related to the global coronavirus pandemic that has affected us all in various ways over the past year. Before we get started, we'd like to know where people are joining us from. If you could please click on the link in the chat window, it'll take you to our polling feature. You can participate in this poll either with a computer or a mobile device. And as you enter your answers, we'll be seeing the results populating on a different screen, and we'll share those results in a few minutes to get an idea of where our audience is tuning in from. We'll give this just a minute for folks to enter their answers. And while we do that, I'll give a brief personal introduction. As I mentioned, my name is Jim Sanford. I'm a native here of the Tri-Cities. I grew up and graduated from Richland High School. And following some time away to attend college at Washington State University and graduate school in San Diego, California, I'm now back. I work as a biomedical scientist at PNNL. So it's really a personal joy for me to be here helping with this seminar series to connect our scientific resources at PNNL with the community. Give it just a little bit more time for these polls to come in. I think maybe as expected, we have a lot of folks joining from the Tri-Cities, Washington area and surrounding areas, Washington in general, but we do have some folks from outside of Washington. We have Portland, Oregon. We've got Annapolis, Maryland, great. Tennessee, Mexico. We've gone international. So we'll share these full results later just to let everybody have a taste of where we're all coming from. But thank you very much for participating in that quick activity. As a quick primer, just wanted to give a little brief introduction. Pacific Northwest National Laboratories, managed by Battelle, is one of 17 United States Department of Energy national labs. We're primarily focused on addressing critical national needs and advancing the DOE's primary missions in areas including scientific discovery, energy independence, environmental management, and national security. With a main campus located in Richland, Washington, and locations in Sequim, Seattle, Portland, Maryland, and Washington, D.C., PNNL provides over 4,700 jobs, a payroll of over $465 million, and over $1 billion in annual spending. You can see some more metrics on this slide. I don't want to take up too much time from the upcoming presentation, but this demonstrates ways in which PNNL is an economic engine in communities across Washington State and beyond. Besides economic impacts, Battelle and PNNL strive to be good stewards in the communities in which we live and work. Battelle has invested millions of dollars, and PNNL employees have logged thousands of volunteer hours each year with local organizations and through outreach activities. One mechanism of outreach is this Community Science and Technology Seminar Series, which has been in existence for more than a decade. Perhaps you've previously attended one of these talks in person or virtually as they've been offered for the past year. If not, I encourage you to keep an eye on PNNL's events page. A link is being provided in the chat for your convenience for information about future presentations. Typically, these talks are given by PNNL scientists, and they focus on the world-class research being conducted right here in our community. However, we've decided to try something a little bit different this time around, and we've organized a 5-part seminar series focused on various aspects of COVID-19 science. As this slide illustrates, we've recruited 5 PNNL researchers who will be giving presentations on the indicated topics. While each talk is beneficial on its own, the seminar series is designed to complement and build upon each talk, and we strongly encourage you all to attend all 5. As an added bonus, if you attend all 5 talks and complete the survey after each, you'll receive a digital certificate of completion. Today, we'll be hearing from Stephen Wiley, who will be giving an overview on what we know about COVID and how we know it. I'll introduce Stephen in more detail in just a moment, but first I'd like to say a few words about the format of the presentation of this seminar series. The speaker of the day, Steve, today, will give a 25 to 30-minute presentation, after which we will have time for some question and answers. I encourage you all to type questions into the chat dialog of the Zoom feature as we go along, and we'll collect these questions and present them to the speaker at the end as time allows. To help spur the Q&A session, we have invited a panel of community representatives from various organizations who will be joining us for each talk. They've come prepared with questions related to the community interests in which they represent. Allow me to introduce our community panel and give them each a moment to say what they hope to gain from today's presentation. We have with us Loanne Ayers, the President and Chief Executive Officer from United Way of Benton and Franklin counties. Loanne, what do you hope to gain from today's talk?
Well, it's a pleasure to be here and learn more about the facts surrounding the pandemic that swept our world. Every day I talk to local small businesses and nonprofits, and we're all struggling with fact versus fiction. So I'm eager to get more information out to the public about what's really behind what's happening and what the future looks like.
Perfect, thank you. Next, we have Kate McAteer, the Vice Chancellor for Academic and Student Affairs at Washington State University Tri-Cities. Kate?
Thank you, Jim. I'm excited to be here to listen to Steve's presentation tonight, and I'm really hoping to learn a little more about ventilation requirements and reducing risks in classroom environments, because we hear of that as being a major concern with faculty.
Excellent, thank you. Next, we have Martine Valadez, the Regional Director for Heritage University's Tri-Cities campus and the Interim Executive Director for the Tri-Cities Hispanic Chamber of Commerce. Welcome, Martine. We'd appreciate a few remarks.
Thank you, and thank you for the invitation. And I'm looking forward, particularly today, to learning more and more about the science behind this. I read a little bit, but I know there's always more to learn about the science behind this pandemic, and also in particular about how kind of diverse populations may be impacted by it and why. Thank you.
Great, thank you. And finally, we have Justin Raffa, the Artistic Director of the Mid-Columbia Master Singers. Justin, what are you hoping to learn tonight?
Thank you, Jim, and good evening, everyone. I have someone that's very close to me who believes that they have had COVID over the holiday break, and I remember one of the first things I asked them was, well, are you sure it's COVID? Maybe it's just a cold. Maybe it's just the standard flu. So I'm excited to learn more about the differences. And I've certainly been overly sensitive these past few months to constantly checking my own body temperature for fear that I've had a little cough or a little tickle and that it might be COVID. So I'm looking forward to hearing from Dr. Wiley to break that down more for us tonight.
Great, thank you. Thank you all four of you representatives for joining us today. Hope we're able to answer some of your questions. Without further ado, it gives me great pleasure to introduce Dr. Stephen Wiley. Dr. Stephen Wiley is a laboratory fellow and biologist at PNNL who builds computer models of underlying cellular processes. More recently, he has focused on using new CRISPR-based technologies to build improved models for understanding cellular signaling and metabolic pathways. Steve, welcome. Please take it away.
All right. Thank you very much for that introduction. And it's a great pleasure to be here to just sort of talk about what we know in general about COVID. Now, my presentation is sort of an overview, getting down to the simple basics of the disease, what we know. And the other 4 seminars are actually going to be expansions. And as we go along, I'll describe about the general things and where we will be talking about certain aspects of the pandemic in more detail. So to start out at the very beginning, first of all, let's say, what is a virus? I think it's really important that we really start and understand what a virus is as opposed to a bacteria. A virus is a non-living particle of genetic material that takes over a cell and forces them to make more viruses. I think everyone has heard about computer viruses, and actually the analogy is actually a very good one. It's primarily genetic material and a particular capsid or coat that allows it to infect certain types of cells. Just like a computer virus will infect certain types of computers and take them over and make them do operations they're not used to, a virus does the same thing. There are many types of viruses. They've been with living things since the beginning, and not all cause disease. Even bacteria have viruses. Now, the important thing, and the reason why it's important to know this is a virus, they need a host to survive. A virus outside of a living host or a host reservoir will not survive for very long. So typically they're transmitted from animal to animal, unlike a bacterial disease, for example, salmonella, which can exist in the outside world without an animal host. A virus needs to come from another host. And we talk about things like host and stuff. So before we go too much further, here's a couple of terms that we will use. I want to make sure that everyone understands what we're saying, because the whole purpose of this is not to, show how smart scientists are, but it's actually to communicate with what we know. And so it's important that you understand some of the scientific terms that we use. A pandemic, as opposed to an epidemic, is an outbreak that's very widespread. Okay, so that's really, an epidemic is a small pandemic, you can say, whereas a pandemic is over a continent, over the whole world. When you say about transmission, we're not talking about power lines, we're talking about spreading a disease. A mutation, I'm sure you've heard about the new mutations that have been described in COVID, is a change in the genome or the genetic material of the organism that may impact transmission, symptoms, or immunity, and it's not quite clear. So a mutation is a general term. Infectious or contagious, that means that you can actually transmit the disease. You can be sick and not contagious, and you can not be sick and be contagious. So being able to spread a virus is distinctly different than actually being sick with it. A vaccine, of course, we know about vaccines. They are a part of the virus or part of the organism that is used as a preventive measure to build immunity against that disease. It's designed not to cause the disease itself, but to provoke an immune response in the host so that the host can fight off the disease without getting sick. And then one thing we'll talk about is models. Models are exactly what you expect. They are a representation of a disease. Anything that you create a model is something that resembles a disease and shows the important aspects of it. Most of the models in scientific research are computer models, mathematical models, computer models, things you can measure, things you can test, things you can make predictions about. And so we'll use this a lot. And finally, a reservoir is any person, animal, plant, or substance which is a source of the pathogen. Okay, so without going too far, let's talk about why we've all come to this talk, and that is not to hear me, but to learn about the virus. This is the culprit. This is called by a couple of different names. 2019 COVID virus, coronavirus, aka SARS-CoV-2. SARS stands for Severe Acute Respiratory Syndrome, and COVID is coronavirus, and this is the second one, there's a SARS-CoV, and this is number 2. They're called coronavirus because this is an RNA. The RNA is the genetic material that they use, has a lipid coat, and it's got these spike proteins. These spike proteins are what the virus used to attach to cells and get inside. You notice that these spike proteins make this sort of this halo or corona around the virus. And that's actually where the name came from, like the corona of the sun, this bright aspect of it. And so when researchers saw this, said, oh, it looks like a sun corona. So they called it a coronavirus. Now, what is so special about coronaviruses? Most viruses are highly specialized for one species. For example, poliovirus. Most people have heard of poliovirus. That actually only occurs in humans. So if you eradicate poliovirus, you eradicate it completely. Coronaviruses are a group of viruses that are RNA-based enveloviruses that originated as respiratory viruses in animals. And they are called zoonotic viruses because they can be transmitted between different species. When this occurs, they can become pathogenic. That is, they can cause disease. Coronaviruses in their native species typically don't cause disease. So they survive with the species and live, and they've been evolving with the species for millions of years. But when they transmit it to a new species, they cause unexpected effects because they're in the wrong place at the wrong time, and our immune response isn't appropriate, and you get all sorts of issues. This is important because coronaviruses have caused more than one disease outbreak. So there's been a number of disease outbreaks or zoonotic diseases from coronavirus, and some of them have come from rats, go through cows. These are historic coronaviruses that have caused diseases in years past. There's a group that we'll talk about here, which come that come from bats. It's caused a couple of them. SARS-CoV is one of the first ones. This virus had a fatality rate of about 10%. So 10% of the people who had this died. The MERS-CoV came from camels, came from bats through camels, and this had about a 30% fatality rate. And here's our COVID-2. And this, we're not quite sure where it was transmitted from. And this has a much lower rate of spread. However, it is far more contagious. These other coronaviruses did not get very far. They were basically localized to local disease outbreaks. But the COVID-19 disease, the SARS-CoV-2 virus, is very transmissible, and that's where we have an issue. So one of the questions that I've heard a lot is, where did COVID come from? Well, one of the first things that happened when scientists identified this disease outbreak is they sequenced the genetic material and compared it with thousands of other genetic sequences that have been collected on coronaviruses from a number of animals. One of the things that a lot of people in the public don't understand is that science is always going out there and trying to find out where the new viruses, where are they coming from? Are they dangerous or not? The World Health Organization, for example, has spent years of following potential outbreaks of disease. And as part of this, we want to develop a database of all potential diseases. So when COVID came out, we had a database to compare it. And we couldn't find an exact match of the COVID DNA sequence, but most of it but the closest was from bats, and based upon some genetic signatures, one of the primary guesses, it came through another animal, maybe a pangolin, before it infected humans. It clearly did not come directly from bats. It had to pass through another animal. Now, one of the things that's important is that there's no evidence that was engineered or came from a Wuhan laboratory. Doesn't mean it isn't. Doesn't mean that it wasn't engineered. Doesn't mean that it doesn't come from. But there's no evidence based on the genome or the transmission pattern. The best guess is that a variant of COVID had been circulating through the Chinese countryside for a couple of years before the outbreak. And it was just one mutation that converted it from being partially transmissible to a very highly transmissible. So why is this virus so dangerous? Well, primarily it's because it's highly transmissible. It's one of the most transmissible coronaviruses that science has seen. The other thing is it can be transmitted well before the symptoms are seen. If you get the flu, you get sick really fast. And typically when you get sick with the flu, you stay home in bed and you don't infect that many people. With coronavirus, or with the COVID virus, you can have it for days before you actually get symptoms. Humans have also not seen anything that close to this virus, and so it provokes a very strong immune response, especially in older people. Next, it attacks important regulatory systems that control blood pressure. So some of the early patients have seen blood pressure going crazy. And it causes severe respiratory issues. Respiratory diseases are serious for older people and those with heart issues. And so therefore, people who have underlying health issues can be severely impacted by that. Now, one of the things that COVID does is exploits what's called the ACE2 receptor system. And it's not important that you know what that is. It's just that this is a receptor that cells use to transmit signals between each other, primarily to regulate blood pressure. This is a very widespread receptor. And so lots of cells in the body have it. It's in your lungs, it's in your corneal epithelium, it's in your nose, it's in your spleen, it's in your liver. One of the reasons that's important is because this means that coronavirus or COVID can infect lots of different cell types. And this means it can cause all sorts of secondary issues. Now, the extent to which this is causing health problems is not quite clear, but the potential is that the coronavirus can really cause a lot of problems in different tissues in the body. Now, when the COVID first broke out last year, one of the questions is, well, it doesn't seem to be any more dangerous than the flu virus. Well, that actually isn't true. Coronavirus is intrinsically more dangerous than the flu for a number of reasons. First of all, COVID symptoms, and there's a lot of confusion about whether it was the flu, as Justin Rafa explained, is that COVID symptoms are similar to the flu, but include shortness of breath and frequently a loss of taste and smell. One of the tissues that the coronavirus can infect is the nasal epithelium, the skin in the nose, or the parts of the nose that we use to smell. And it's thought that that it induces an inflammatory response, and you can't smell. Flu does not do that. Flu symptoms include headaches. Coronavirus typically doesn't cause headaches. And it also typically doesn't cause the aches and pains that you have typically with the flu. So if you're feeling your head hurts and you're achy all over, it's probably the flu. If you can't smell anything and you can't breathe and you're coughing and you feel like there's something wrong with your lungs, it's probably coronavirus. Now, early in the pandemic, one of the things that was the most striking about it is the fatality rate is strongly biased by age. And this was quite surprising. This is an analysis of hundreds of thousands of cases that was accumulated early last year. And as you'll notice, if you look at the age of the subjects and look at the fatality rates, if you were greater than 80 years old, you were talking about almost a third of the people were dying. But young people were not showing, even though you had lots of people who had been infected when they were young. And you can see that the infection rate peaked at 50 to 59, probably because of the people who were exposed to it. These people did not show such a death rate. This was surprising because you've probably heard of the influenza epidemic of 1918. And that was, killed millions of people around the world. It primarily affected young people. So the children were dying, but the old people were okay. This is the opposite. The young people can get it, they can transmit it, but they seem not to suffer as severe an effects as the older people. Now, research since this data came out indicates that a large fraction of the people who are dying is because they mount an excessive immune response to the virus. So what scientists started doing, or doctors started doing, is to treat the patients with anti-inflammatory drugs so that they don't over-respond to it. And in fact, scientists have found that if you go into the bat populations, one of the ways that coronaviruses and bats have coexisted for so many years is that the bats do not mount a strong immune response to it. And so now they've been able to bring this death rate down by using drugs that suppress the inflammatory response, like hydrocortisone. And I think that this will be covered in one of the talks. I think Amy is going to be talking about that in the next talk. And small children, they get sick, but it's probably they don't get quite so ill is because they don't mount such a strong immune response. So you can imagine when these numbers came out, scientists said, this does not look like what we're used to. And so they realized that this was something new that we had not seen before. So what did they do? Well, scientists, the first thing we do is we build a model. And that's what scientists do, because you want to explain it. It's a complex, it affects people, it's different, it has different transmission, it has different sensitivity. And so therefore, it's really important that we understand what's going on. And so this is just an overview, and we'll be talking about different parts of this model in the other 4 talks in this series. And what the model does, it simply shows different pools. This is the susceptible people. These are the exposed people. These are the infected people. These are the infectious people. And when we build these, we basically take every potential possibility and we draw errors between them. And we say, okay, susceptible or exposed at what rate? Then you have to get data for that. And of the ones that are exposed, how many get infected? And you have to measure that. So you can build a model, but until you get the numbers, which really describe how these different pools shift and how these different processes operate, this was really sort of uncertain. So the early models that we built, when you can consider the models sort of similar to, there's a lot of models that we build on all sorts of things. These original models were based upon the historic experience with other coronaviruses and other types of infectious disease like flu. But we understood right from the start that there would be a lot of uncertainty. And so and this gives an example that when scientists at the University of Washington were trying to do projections, and this shows how the projections change very quickly. This is a projection in April 2nd. By April 5th, they had changed a little bit. By April 7th, they had changed it a bit. And when they made these projections, there was these uncertainties. And they say, well, it can be this or this, and this is the uncertainty. And the uncertainties basically reflect the lack of data that we had early in the pandemic about what those parameters were that we had to worry about. And that's why very early in the pandemic, everyone said, get tested, get tested, get tested. Even if you don't think you're sick, you get tested. And there was a burning desire to have as much data as possible so we could construct realistic models of what was going on. But one of the things that was very clear from the early parts of the model is that there was a rebound. So if you just take the general model, and let's say you do this one, if you say, okay, at the epidemic growth doubling time of 4 to 7 days, this was what the model said. If no one does any social distancing and everyone just goes about their business, you get a peak of infection, lots of people die, then it goes away. End of story. Let's say that you now have social distancing to flatten the curve, then would go up and it would come down. But at some point, basically, if you look at the area underneath this curve, you just have as many people here as you had here. They just take much longer to get impacted. And actually, one of the things that really drove the original lockdown and the social distancing efforts was the fact that 30% of the people the older people who were getting the disease were dying. And so the doctors were scrambling to find some effective way of treating them. Let's have an antibody drug. Let's have an antiviral drug. Let's try lots of things to lower the death rate. And that would take time because you just can't, invent things instantaneous. So a lot of what was done early on was to delay the disease so that we could find out effective treatments. But one of the risks they all knew is the fact that if you, for example, if you look at this teal line here, if you basically said, okay, let's social distance, let's mask, let's stop, and then drive down the risk to a certain point, and then if you remove social distancing and everything goes back to normal, then you're right back to where you began and you really don't get many benefits. So one of the things that we were hoping for, or that science was hoping for, is the fact that before you got this rebound, we'd have a vaccination, we've had effective treatments and things like that. This fear of the rebound was obviously well-founded. If you look at the original pulse, and you can see here that in June, during the Memorial Day holiday, people stopped social distancing and bingo, it went back up again. It went back up again. Spain did something very similar. We were delayed from Spain, and you'll notice that different countries had different progressions in the middle of last year. And it was very clear if you analyze the slopes and when these things came up and matched from the mathematical models, most of these rebounds had to do with people going from distancing and mask to non-distancing. And so this was an issue. And of course, we still continue to fight with that because, as you'll notice here, that original graph was taken about here, and you can see what's happened in the meantime. This is in Europe. And we were talking about here in July when you had the rebound. Now we had this huge rebound, which was apparently driven by things like Thanksgiving and Christmas and things like that. But the good news is that the death rate did go down because during that delay, we were able to find more effective treatments for the older patients. And so the number of people who died from COVID as a percent of the infected definitely went down. Now, one of the most important questions that everyone asks me is, when will it end? Well, this pandemic will end either until everyone is infected or most people are vaccinated. I mean, that's the only two ways. And this was very clear from the beginning. COVID is unlikely to disappear on its own because it's very infectious. If it's around, it will find people. So early lockdown measures, as I said, was to delay infections. And now we're not dying, but developing an effective vaccine is the end game. So until you get vaccinated, the best way to reduce infections is to reduce the transmission. And as I said before, because the pre-infection phase of COVID-19 is so long, most transmission is likely to come from people who don't know they have the disease. Transmission is mostly through aerosols. At first, they wasn't sure whether it was going to be through aerosols or through contact to surfaces. And after a while, it was clear that it was aerosols that was really transmitting the majority of it, which means if it's indoors, good airflow is essential for reducing risk. You want to reduce the concentration of the aerosols, and I'll explain in a minute. And the best way to reduce transmission is by social distancing and wearing face masks. Now, the reason why social distancing is so important is because of the logarithmic or the exponential aspects of how people get infected with these kind of diseases. Because it's so transmissible, you could show that if you have the normal behavior, you could show that one person not changing their behavior in 5 days would infect 2.5 people. In 30 days, one person gives rise to 400 infected people, which is why these small outbreaks or these events, what they call these super spreader events, in which 1 or 2 people were infected and suddenly it transmits very quickly. If you simply lowered the contact by 50%, instead of having 406 people in 30 days, it's down to 15 people. If you reduce it to 75% less contact, you're down to 2.5 people. So you can see that you don't have to get the transmission down to 100% to really make a significant difference. It does make a difference by just doing these small steps. And the reason is because coronaviruses, why distancing and masking is so important, is because coronavirus was actually designed to be transmitted by the air. And that is, if you're a bat, remember it evolved in bats. If you're a bat and you're hanging upside down in a cave all day and then you fly out at night, how do you transmit a disease? Well, you scream. I mean, that's how bats locate their prey. They basically use their echolocation to find their prey, which means they expel large amounts of sound. And apparently the coronavirus has used that as a way of transmitting. And so that's why when there was no surprise when the original studies about transmission showed that a lot of the transmission was taking in bars and exercise rooms and from singing. And so I used common sense to avoid that. Now I use the term airborne transmission. Infectious aerosol particles can be released during breathing and speaking. These are not the virus particles themselves. These are small water droplets. If you've ever spoken in an excited way, you know that you can almost spit. And the virus hitches on these particles. And if you wear a mask, what you're doing is you're preventing the transmission of those small particles. They're not perfect, but if you're wearing a mask and other people are wearing masks, the transmission is really very, very low. Now, I said that the end game is developing a vaccine, and frankly, the good news is that new approaches to vaccine development have been in the works for many, many years. Classical platforms were based, like the development of the polio virus and the measles virus, were based upon centuries of work. Well, over the last 20, 30 years, genome-based science or sequencing genomes have allowed us to really understand at a fine level of detail how these things work and to devise very clever ways in that you don't need the whole virus itself, you just need the genetic material. So some of the new viruses are like to take this genetic material, put it in a harmless virus and infect people. This is the Oxford AstraZeneca one. Or you can take the RNA to make mRNA, just like the RNA in the virus, and then put that in a lipid droplet and inject that into cells. And if you look at these are the two vaccines that are available here in Washington State. This is the one that's available for the most part. And I think this is the new Johnson & Johnson virus is similar to this. And so these are very very, very efficient. A lot of them were developed based upon basic scientific research to try to fight not only pandemics, but bioterrorism attacks. And so there was a lot of research going behind the scenes for many years. And so when the pandemic came, it just gave us scientists an excuse to go out and try those cool ideas that we had been playing around with for a while, but didn't have a chance to try out. So that's my overview. And so from susceptible to exposed to infected and infectious. And so what we'll talk about in the meetings, the next thing is various parts about the immunity, the mutations, the infections, how to stop it, how to stop transmission in various parts. So thank you. If you have questions, submit them versus the chat. And thank you for listening and attending this talk.
Great. Thank you, Steve. Thank you so much. We all appreciate your time here. Appreciate you taking the time to discuss this topic, and I know that everybody in the audience appreciates it as well. We have more than 250 people joining us tonight, so that's a wonderful number. And there's been quite a few questions that have percolated through the chat. I'd like to jump right into the Q&A portion. As I mentioned at the start of this, we're going to lead with some questions that are coming from our community representatives. So let's start at the top. Loanne, do you have any reflections on Steve's talk tonight and a specific question you'd like to ask based on what you heard?
Thank you. It was really informative. I think we're all focused on the slide that said, when will it end? So, there are really two complex scenarios affecting when will it end, both vaccine availability
Mhm.
and acceptability. What are the key success factors for each?
Well, frankly, right now a lot of people were afraid that that people were not accept the vaccine. So, for example, there are certain populations that, oh, we'll never take a vaccine. So far, uptake has been very, very high. And so, as we know, we can't. I'm eligible to get the vaccine. I have to wait. So I think that if you look at the demand and look at what is required to really reduce the transmission, I don't think that people's reluctance to get the vaccine is going to have a major impact. Remember, they don't necessarily have to be vaccinated. They either be vaccinated or they get sick. That works too. I'd rather get vaccinated myself. Does that help?
Thank you.
Great. Thanks, Loanne. Let's move on to Kate. Kate, do you have any thoughts and questions for Steve?
Yeah, thank you, Dr. Wiley. That was an excellent overview. So as you know, lots of people want schools to reopen and get our kids and our students back in the classroom. And so you talked about masking and distancing. Are there other safety considerations that those of us in education should be considering as we reopen our classrooms?
Yeah, it's sort of like any kind of exposure. Think about it's not just am I exposed? It's how long am I exposed? How close am I? Do you talk to each other? For example, the point has been made that movie theaters are much safer than restaurants because people are talking are all facing the same direction and they don't talk. In schools, for example, you can set up the school with masks so people that are facing in the same direction and wear more effective masks. There's some masks that work much better than others, and reduce the exposure time. So it's a matter of just time, exposure, dose, concentration, good air circulation. If you get the risk level down low enough, it's pretty safe. And remember, it's not just the risk of of catching the coronavirus. It's also the risk of not going back to school that you have to weigh. And that's something that scientists can't do. We can only tell you what your risk is for the coronavirus. It's other policymakers who have to weigh that risk. But if you get it low enough, I think that you can make people comfortable enough to go back.
Thank you.
Great. Thanks for the question, Kate. Thank you, Steve. I'm going to move on to Martine. Martine?
Thank you, Dr. Wiley, for a great presentation. One of the concerns for some folks is the reactions or some negative that some folks have to the vaccine. And I was wondering if you could speak to that a little bit.
Yeah, I think that they'll cover it a little bit more in the future talks. But I will say that if you don't get any reaction at all to the vaccine, it doesn't work. Because what you're actually doing is you're getting an immune response, but it's supposed to be a small controlled immune response because your body is actually reacting to the vaccine itself. And so one of the things about these new synthetic ones, the Modena, the ones that we're getting here, these are synthetic vaccines. So the synthetic vaccines typically have lower side effects because people haven't really seen this kind of substance before, and they're engineered to get a low response. So typically the new vaccines have much lower negative response rates than the older ones did. Now, having said that, yes, there's always the second shot I hear is terrible, but not as terrible as the shingles vaccine. And so it's like you pick your poison. But typically there have been, compared to other vaccines, the adverse reactions have been very low.
Great. Okay, we'll move on to Justin. Justin, anything in particular you'd like to ask?
Dr. Wiley, just earlier today, the governors of Texas and Mississippi announced lifting their state's masking requirements and are allowing all businesses to return to operating at 100% capacity with no restrictions. In your opinion, is this an appropriate time for state governments to lessen, or in this case, eliminate these COVID safety protocols?
Well, it depends upon whether or not you're trying to minimize the transmission, or you're trying to maximize the economic recovery of the country. And that's a policy issue. I can say that from a scientific viewpoint, the probability is very high that their number of COVID cases will rise significantly. And based upon historical data, the death rates will go up and other secondary things. Now, all scientists can do is tell you what the likely outcome will be. It's up to the government to weigh that and make a decision based upon that. Now, if you ask my personal opinion, not my scientific opinion, I like my life, really. But hey, you know, I'm not shut down, so you know, I'm the wrong person to ask that.
Very good. Thanks, Steve. Thank you to the panelists' questions. We have quite a few questions that are coming in through the Zoom chat, so I'd like to hop over to a few of those. I would first like to point out that there is 4 more talks in this seminar series, and some of them are more directed towards things like the vaccines. So a few of the questions that have popped up are certainly going to be answered in the upcoming talks. So if your question is not answered tonight, you know, there's a little bit of a time issue, but if your question is not answered tonight, I hope that some of the next 4 talks will help address those. But from the chat, we're getting a few questions related to, do we have any idea of why some people are asymptomatic or have very mild symptoms?
Well, it may very well be because they have had a prior exposure. Like, for example, one of the things that was early in the pandemic, they would do antibody tests, and they would see people with antibodies that didn't get sick. For example, on cruise ships, on AV ships, in which you had a lot of people together, they tested them. Some people had the sickness and had antibodies, of course. Some people had antibodies, but not the sickness. And it's thought that they got a small exposure, not enough exposure to get them sick, but enough exposure to generate antibodies. So that it's a matter of, as I said, it was a matter of time, exposure, and dose. If you get a large dose, you'll probably get sick. If you get a small dose, you may not get sick. You may develop antibodies in a mild case, because it's a race. The COVID is trying to infect your cells. Your immune system is trying to fight it off, and who wins that race depends upon whether you get sick or not. So that's probably the reason why.
Great. Kind of in line with that, in terms of people's different reactions to the virus, do you have any insight into what factors might contribute to certain individuals eliciting a strong autoimmune response after being infected?
That's a great question. I wish we knew the answer to it, because in a sense, that's what seems to be so different about this particular coronavirus. It really elicits a very strange immune response. It seems to look like an autoimmunity. It may be the cells are fighting the ACE2 receptor. I don't know. It's sort of hard to tell, and so, but a lot of scientists are very, very interested in this as basically as a way of investigating the immune system. One of the things that's not appreciated is that science spent many years developing a vaccine for the HIV, AIDS. And AIDS basically attacks the immune system. So the first thing that AIDS does is it knocks out your immune system. And so a vaccine doesn't work. But science spent an enormous amount of time and effort understanding how the immune system worked, how to react to HIV, how they could get around it. And that basic science led to a lot of insight that we use to fight the coronavirus. And so people say, how in the world did you develop a vaccine so fast? Well, we've been working on vaccination of a bunch of diseases for decades. And finally it was like, hey, it worked, fine. So we'll expect that finding things like this long-term chronic COVID, which looks like an autoimmune disease. Investigating that will tell us a lot about the immune system, may tell us about autoimmune diseases, may actually lead to new treatments. That's the way that science works. You learn in one area and you apply it to another area. So I'm hopeful that we'll learn something from that.
Me too. Great. So we're hearing a lot these days about the different genomic variants of SARS-CoV-2. Some recent studies show that in certain areas, Houston, for example, there might be more than 5 different variants circulating around at a particular time. Do you think that this virus is evolving abnormally fast or slow? And how does that impact where we're headed?
Evolution of a virus is related not only to the frequency at which it develops a mutation, but also how fast it's replicating in the pool. The very fact that COVID has infected so many people gives it lots of opportunities to develop a mutation. If you look at per infection, the mutation rate doesn't look to be particularly high. In fact, I think it's relatively low, certainly compared to things like influenza virus, which loves to mutate, which is one of the reasons we have to have new vaccines every couple of years. The problem is the fact that as long as it's widespread and spreads throughout people, the problem is not only that a lot of people are getting sick, even if they don't die, but they're providing lots of opportunities to develop new variants that may be much more transmissible and more deadly. And so you're playing a risk. So far, none of the mutations have really made the vaccines non-functional. They've reduced it because the mutation has to be where the vaccine is attacking. The good thing about the new synthetic vaccines, the Modena vaccine, for example, it's designed on a computer. So basically you say, mutations, and they type in on the computer, here's 10 mutation, push a button, synthesize a new vaccine, voila, you've got a new vaccine. And so I don't worry about it so much because of the properties of the coronavirus and scientists' ability to alter the vaccine. As long as we can keep the infection rate down to a certain point, it won't be a problem likely.
Excellent. Any idea if there's any specific data related to poor air quality, poor ventilation, high pollution areas, and how that can affect the severity of COVID-19 in patients and their ability to recover from disease?
Yeah, this has to do with the comorbidities or pre-existing conditions. One of the things that was an idea that was pursued early in the pandemic was the fact that some of the death rates in China were associated with people living in densely populated cities with air pollution and with lung conditions. So if you live in an polluted environment, your lungs aren't as healthy and you're more likely to die of a respiratory disease. I mean, it's that simple. And so yes, if anything that will compromise your lungs or your heart or any of the systems that are attacked by coronavirus put you at greater risk for disease and bad outcomes.
In line with that, is there any idea about molecular causes of long-lasting fatigue and some of the long-haul symptoms that are associated with, that some patients have for weeks and months after the virus is no longer detectable in their body?
People are studying that. The National Institute of Health has recently announced a major program to study it called Long COVID. And they're recruiting proposals from people. I think there's some people at P&L who are interested in doing this. And this is to understand why this is happening. My best guess from just looking at the literature, I'm not an expert in this area, but my best guess is the fact that the COVID is promoting an autoimmune response. Now, against what, how, what cell types involved, they don't know. But that seems to be that the body thinks the COVID is still there, even though it's not, and it keeps mounting an inflammatory response. But obviously, the National Institute of Health is setting up a major program to study it. If I, they don't know. That's the whole point. So wait, stay tuned, and I think we'll find a lot more out in the future.
Great. Okay, I think we've got time for maybe one or two more questions. I know this is one that's at the top of a lot of people's minds, but can you still get sick if you've had the COVID-19 vaccine, or if you've already had the virus? Do you still need to take the same social distancing, masking measures as before?
That's a good question, and it's unclear. What they have found, if you have a vaccine, I think they found some people who have gotten sick, but the severity is very low. In other words, they don't die. They're very low death rate. If you're vaccinated, the death rate basically drops to almost nothing. You can get partially sick. It depends upon how well your immune response responds to it. If you are vaccinated, you can still transmit, but probably not as much as before. So the idea is, if you've been vaccinated, until we know how much you can transmit, it's better to be kind to your other people and wear a mask until everyone is vaccinated. Then if everyone is vaccinated, your transmission is low, your potential to be infected is low, you won't get sick, and they'll be like, I have a cold. The thing is, you're not going to get rid of it. And people will say COVID is here to stay, just like the common cold is, and the flu. And the flu, 60,000, I think, people die from it every year. So it's not like a trivial thing. But the nice thing is that we want to turn it into a low impact, manageable disease. Diseases in humans have always been there. We're not going to get rid of disease, but if we can make it manageable, then I think that people will don't worry about going out to a restaurant or going out to a live performance, and then we can get our lives back to normal. So that's what we want to do. Reduce the risk down to a point where people are comfortable resuming their lives.
Excellent. Thanks. Well, we're coming up towards the end of the hour. There are tons of questions that I wish we could stay around for another hour just to get to. I personally am hungry, and I'm sure others are. It's towards the end of the day, so I think we're gonna stop here. There's lots of thank yous in order. Steve, really want to thank you for taking the time for your overview and sharing your information. Really want to thank all. There's 216 people on the call right now. I think at top number I saw about 259. So thank you everyone for tuning in. Let's crack 300 next week. That'd be great. I want to thank our panelists, Loanne, Kate, Justin, and Martine. Thank you guys for attending today. Really looking forward to hearing from you next week as well. Note that everybody has busy schedules. 5 to 6 is a tough time depending on what you have going on, but really appreciate everyone joining today. Everyone that called in, one more reminder to take the survey that should be coming up in the chat box now, and I think when you close Zoom it should pop up as well. You can also sign up to receive notifications for the future seminars at the PNNL events website. So this will be the next 4 Tuesdays. So every Tuesday in March, we'll be having a subsequent COVID-19 seminar. I really hope that everyone can join us, recruit your friends. This was very informative, and the next 4 will be as well. And yeah, so next week, March 9th, we're going to hear from Amy Sims, a biomedical scientist here at PNNL. The title of the talk is What Do Bats Have to Do with It? Same time, same place, 5 p.m. Tuesday here on Zoom. There'll be a registration link in the chat window and via email. And if you need any more information about this seminar, again, this was recorded today, so a recording will be posted in the coming days. And future seminars, that's all on the PNNL events website. The link has been provided in the chat. And again, thank you very much for attending today. It's been lovely to be here with you, and I look forward to next week. Thank you. Have a nice night.
Thanks for attending, everyone. I appreciate it.